Okay, so last time we started looking at carbohydrates. Started looking at carbohydrates. So you can check out the video for further information on what we've looked at so far. However, we said that our objectives for the lesson on carbohydrate is for us to be able to understand the functions of the carbohydrates, for us to be able to distinguish what a monosaccharide and what a disaccharide is, and what a polysaccharide is. And also we need to describe a sugar, structure of the sugar, and also to name the different forms of carbohydrate molecules, and also to relate the structure of polysaccharides to their functions. And then we need to compare polysaccharide of storage function and also those that are of structural functions. So carbohydrates are part of what are called the macromolecules. We have the proteins, carbohydrates, lipids, and the nucleic acids. And so we are still concentrating on the carbohydrates. So we said that carbohydrates are mainly divided into monosaccharides, such as glucose. Then we have disaccharides, such as lactose. And then the other main division is polysaccharides. So there are different types of polysaccharides, such as starch, chitin, cellulose, glycogen, and others. So we have polysaccharides. Then we could also have a grouping which is called the oligosaccharides. So the oligosaccharides usually have monomer or units 2 to 10 monomers. So we defined what carbohydrates are. We said that they are hydrates of carbon and reflects the ratio of 2 to 1 in relation to hydrogen to oxygen, which is the same that is found in water. We said that these molecules have a carbonyl group and several hydroxyl groups. Said that these are carbonyl group and multiple hydroxyl groups provide sugars with an array of reactive and hydrophilic functional groups. And we said that carbohydrates are either aldehydes or ketones. So either they contain an aldehyde group or a ketone group. So an aldehydes, these are they that contain a terminal carbonyl group, and these are easily oxidized. So mark that word, they are easily oxidized. The aldehydes are easily oxidized than the ketoses. The ketoses have a non-terminal carbonyl group or an intermediate carbonyl group. Because of these aldehydes being easily oxidized, they act as reducing agents. They act as reducing agents. So then we mentioned the functions of the carbohydrates being energy production or immediate energy provision, energy storage, building macromolecules, structural support, sparing protein function, lipid sparing function, and prevention of ketosis. Also said that they play an important role in cellular recognition processes. So we're going to look at these functions or realize these functions as we look at the individual carbohydrates. So when you look at monosaccharides, mono simply means one. So these are the simplest sugars, starting with those with 3 carbons. So those that are having 3 carbons are what are called trioses. Then we have the pentose sugars, which are those that have 5 carbons. So we have an example of ribose and ribose sugars, which are found in DNA and RNA. So ribose sugar in RNA and deoxyribose sugar in DNA. Then we have the hexoses, which are glucose, fructose, and galactose. So the other important thing we mentioned that sugars, especially the pentoses and the hexoses, in nature or in solution, do not appear as straight chains. They appear in their ring forms. So they appear in their ring forms. So you're going to see how they form the forms, those ring forms. So we looked at the trioses, which are the 3 carbon. They have 3 carbon atoms. So I said that conventionally the numbering of these carbohydrates starts at the carbon nearest or at the carbonyl group. So this one is going to be carbon 1, 2, 3. For the dihydroacetone, this will be carbon 1, carbon 2, and carbon 3. The pentose sugars, said we have a ribose sugar and a deoxyribose sugar. The ribose sugar is a component of RNA, or the deoxyribose sugar is a component of DNA. Then we said that the difference is in carbon 2, where we have a hydroxyl group in a ribose sugar, while just an oxygen atom in the deoxyribose sugar. And so we also said for the molecular formula for a ribose sugar, we see that it has C5H10O5, which is in relation to the general formula of carbohydrates of CH2ON. But when we see the deoxyribose sugar, it has C5H10O4. So C5H10O4. So it kind of deviates from that general formula. So I said that for the ring formation of ribose rings, carbon 4 and carbon 1 are going to react. So the carbons, the positions of the carbons that are react, or the reaction takes place between carbon 4 and carbon 1 to form these furanose rings. So today we're going to begin on the hexose sugars. We're going to begin with the hexose sugars. So before we go into the hexose sugars, when we look at the levo and the dextro, levorotatory, dextrorotatory. So we find that when we look at the D-glyceraldehyde, which is a triose, it's a triose. When we look at a D-glyceraldehyde, which is the simplest carbohydrates, this in a D sugar, we find that the hydroxyl functional group, is on the right side of the asymmetric carbon atom. So in this case, we have the asymmetric carbon, Now that asymmetric carbon, the hydroxyl group on that asymmetric carbon will determine the side, will determine whether a sugar is a D sugar or an L sugar. So in a D sugar, the hydroxyl functional group is on the right side of the asymmetric carbon atom. So when you look at the D-glyceraldehyde, this carbohydrate, it rotates light to the right, as we said last time. So this glyceraldehyde was actually, or is the one which is actually used as a reference to categorize a sugar as a D or an L sugar. So it was the one which was chosen. It was conventionally chosen as a reference point. So now coming to the hexoses, the hexose sugar. So these are 6 carbon sugars. So they have 6. So hex means 6. So we have, remember, we're starting counting from the carbon at the carbonyl group or nearest the carbonyl group. So we are starting counting 1, 2, 3, 4, 5, 6. This is glucose. So glucose has C6H12O6. When we look at fructose, it still has the same molecular formula. When we look at galactose, it still has the same molecular formula. When we look at fructose, however, we find that fructose is a ketone, is a ketose, so it has a ketone functional group. Or when we look at glucose, glucose has an aldehyde group, and galactose also has an aldehyde group. However, when you look at glucose, we find that glucose and galactose find that they differ in carbon 4. The position of the hydroxyl group. The position of the hydroxyl group. Remember, we looked at isomerism. Isomerism. So from the concept of an isomerism, what type of an isomer is glucose and fructose?
I think stereoisomers.
Stereoisomers, glucose and fructose. How about glucose and galactose?
Oh, I thought it was glucose and galactose.
So you are saying glucose and galactose are stereoisomers?
Yes.
Okay. How about fructose and glucose? Remember the functional group. One is an aldase, the other one is a ketose. Yeah, so you're right. So glucose and galactose are stereoisomers, while fructose and glucose are structural isomers. So remember that fructose and glucose are structural isomers. Glucose and galactose are stereo isomers. Thank you for that. Okay. So now when we look at these are hexose sugars. So we find that the hexose sugars are actually very important direct source of energy. So during cellular respiration, they are oxidized. They are oxidized. And the most common respiratory monosaccharide, or the most common hexose or monosaccharide that is used during respiration is glucose. So glucose is the most common respiratory monosaccharide. Then we also find that these hexose sugars, they are used in the synthesis of disaccharides, as we are going to see as we continue, and also oligosaccharides, and also polysaccharides. So they are very important. They are the most important source of direct energy, actually. So now, glucose is actually the most abundant monosaccharides and is used as an energy source in most of the organisms. So I find that during respiration, cells are able to oxidize the glucose molecules. So that energy that is stored in this glucose molecule is able to be used by the cell for its function. Then we also note that glucose is also used in the synthesis of other types of compounds, such as amino acids and acid, just as we looked at or explained when we looked at the functions of carbohydrates. So now we find also another important property of glucose is that it is soluble in water. Remember that glucose has hydroxyl groups. So those hydroxyl groups are able to form hydrogen bonds with water molecules. So they are able to form hydrogen bonds with water molecules. Remember I showed the open chain for form of glucose, and it has an aldehyde group. So for them to form a glucose pyranose ring, remember we said that in the ring form, the carbon that contains the carbonyl group, the carbon at the carbonyl, the one that forms the carbonyl group, will react with one of the carbons with an hydroxyl group. So we expect that for the ring formation of glucose, for a ring to form, we expect that carbon number 1 will be part of the reaction, and then also carbon 5. So carbon 1 and 5 will combine to form a 6-membered pyranose ring, to combine to form a 6-membered pyranose ring. And then the other property is that glucose is a powerful reducing agent. It's a powerful reducing agent because it has a presence of an aldehyde group at carbon 1 in the open chain. So when you look at the open chain here of glucose, carbon 1, this is carbon 1, right? So we find that this aldehyde group, remember I said that aldehyde group is easily oxidized. So this makes the glucose to be a powerful reducing agent in its open chain. So it's a powerful reducing agent. Okay, so this diagram is showing the synthesis of or the formation of the ring form of glucose. Now, before we look at that, remember I talked about a reducing sugar. So we find that in a reducing sugar, there are tests that are done to determine whether a sugar is a reducing sugar or a non-reducing sugar. So we have tests such as the Benedict's test and the Felling's reagent. So we find that this test will give a color change when a metal is reduced to the metal itself or to an ion of lower oxidation state. So we find that the reagent will oxidize the sugar, or the sugar is reduced, or the sugar reduce the oxidation state of the ions. So for a reducing sugar is any sugar which reacts in basic copper 2 ions solution, which yields a copper oxide or copper 2O, so Cu2O, precipitate. So this is doing the Benedict's test. So a reducing sugar is going to react in basic copper 2 ion, and then it's going to yield copper oxide precipitate. So these sugars are those that contain the aldehyde group that can be oxidized, so that they are oxidized. However, we find that all monosaccharides are actually reducing sugars. So even fructose, which is a ketose, is also a reducing sugar because it's able to isomerize to glucose. It's able to isomerize to an intermediate with an aldehyde group. So it's able to a process called tautomerize. So it's able to tautomerize and isomerize into a molecule with an aldehyde group. So we also find that all the common disaccharides, except sucrose, are also reducing sugars. So we find that lactose, maltose, cerebiose are reducing sugars. So disaccharides, except sucrose, are also reducing sugars. So when we look at the polysaccharides are generally not reducing sugar. So they are generally not reducing sugars. So coming to the formation of the ring formula, or form rather, the ring form of glucose. We said carbon one will react with carbon 5. So this is the first part showing the orientation of as a ring is about to form. So we have carbon 6 there, we have carbon 5. So remember, it's carbon 1 and carbon 6 that is going to react. So here we have this is a covalent bond being formed between this oxygen atom and the hydrogen. So here this bond shows a sharing of electrons. So this is a pair of electrons. There are 2 electrons. So what will happen is that these electrons, remember the arrow is showing that these electrons are coming to the oxygen atom there. And then this means this hydrogen will be H plus. It will have the configuration of H plus, or a proton. And then this oxygen will react to form, or react with this carbon to form the covalent bond, this covalent bond, this covalent bond rather. And then this, sorry, this oxygen reacting with this proton, this hydrogen ion, will lead to the formation of the OH there. So of importance is for you to know that the reaction takes place between carbon 1 and carbon 5 to form a pyranose ring. So depending on the position of the OH group, so the OH group, if it's down below the plane, this will be alpha. If it is below the plane, it's alpha. It is above the plane, it's beta. So this one, OH below the plane, down there, this is alpha. Above the plane, it's beta. Below the plane, alpha. Above the plane, beta. Then we have galactose. Said galactose is a stereoisomer for glucose. So galactose is a water-soluble monosaccharide, and remember, it's also an aldase, just like glucose. Remember, I said that the difference is on the OH group on carbon 4. So the OH group in glucose is below the plane, while in galactose is above the plane. So we see that when these molecules form the pyrrhose ring, they're going to differ, because at C4, where the OH group is above the plane in galactose, in glucose it is below the plane. So remember, you're looking at C4, carbon 4, carbon 4. So glucose, it's below the plane here. it's below the plane. So glucose below the plane. Then galactose above the plane. The other thing that we need to note is that galactose will combine with glucose to form a disaccharide that is called lactose. Lactose is also commonly known as milk sugar. Milk sugar. So lactose is present in milk. No wonder it's called milk sugar. So galactose, a monomer galactose, a monosaccharide galactose plus glucose will give us a lactose. And then we also note that galactose is a powerful reducing sugar because of in its open chain, it has an aldehyde group. So it's a powerful reducing sugar because of it having an aldehyde group. Galactose will also form a pyranose ring. So similarly to glucose, carbon 1, where there's a carbonyl group, N carbon 5 will be involved in the reaction. Carbon 1 and carbon 5. Then remember I said galactose is a stereoisomer for glucose and a diff on carbon 4. So remember counting, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4. So at carbon 4, in galactose, the OH is above the ring, the plane. It's above the plane. Remember in glucose, it was below the plane. The OH was below the plane. So this is the formation of galactose from this pyranose ring. Then the other hexose sugar is fructose. So fructose is commonly, the other name for fructose is fruit sugar. So it's a water-soluble monosaccharide and is found in honey, fruits, flowers, berries, and also in root crops. And remember, in the open chain of fructose, as we saw earlier, we said that fructose is a ketone. It has a ketone. So it means that the carbon 2 of fructose will be the one to participate in the reaction to form the ring form of fructose. So carbon 2, since that's where we have the carbonyl group, will react with the OH group of carbon 5 to form the 5-membered furanose ring. So just as fructose, we find that just as glucose, fructose is also used as a direct source of energy in cellular respiration. Then of importance is that fructose is a weak reducing sugar. Because remember, fructose has a ketone carbonyl group. It is a ketose and it is not an aldehyde. So no wonder it's a weak reducing sugar. What makes it a reducing sugar is because its open chain structure is able to isomerize, as I explained earlier. It's able to isomerize into an aldase sugar. So it's able to isomerize into a glucose structure. And remember, glucose is a powerful reducing agent. It has an aldehyde group. So no wonder fructose is a reducing sugar, but a weak sugar because it has a ketone group. So carbon 2 of fructose is asymmetric. So it's going to lead to the formation of alpha and beta isomers, just like those we find in glucose. And remember, I said that fructose is a structural isomer of glucose. So the formation of the furanose ring. So remember, I said it's carbon 2 because the carbonyl group is at carbon 2. And for the ring form to form, we said the carbonyl group, the carbon at the carbonyl group, that will be involved together with a hydroxyl group. So for Fructose, it's carbon 2 and carbon 5. So this is simply showing the reaction here that will take place. So we have carbon 5 and carbon 2 being involved in the reaction to form this furanose, fructofuranose ring. So this is alpha because it's below a ring. Alpha because it's below, the OH is below the plane, it's below the plane, so it's alpha. If it's above the plane, then it could be beta. Okay, so it simply is showing the same thing. OH of carbon 5 reacting with the carbonyl group or the carbon at the carbonyl group. So position 5 and position 2, which is going to lead to the formation of fructofuranose ring, the furanose ring. So if it's below the plane, this is alpha fructose. If it's above the plane, the OH above the plane, it's beta fructofuranose. So alpha, beta. Okay, do we have any questions before we look at the disaccharides? Any question?
No, sir, it's clear.
Okay, so we can just log out and then log in since it's almost time.